like this one. I mean, like some of the amazing parts.
Yeah. Yeah, and then deep MRI is basically the main project that we're working on now, that we spend most time on. And we just presented it at the Vision Sciences Society, at the annual meeting of the Vision Science Society, VSS. And I felt it was received very well, and people liked it, liked the overall idea, and we ourselves liked the idea a lot. And I personally think this is actually a really important topic, because it's basically a new eye tracking framework. It's also why we spell it deep MRI as EYE. So for the listeners, Benjamin just asked me to spell it. Just
yeah, exactly.
Exactly.
I feel like that was half of the attention on Twitter. It was just the name.
It was the name, yeah. Like, I mean, seriously, I think the name and overall the package as a whole, of course, helps to get the message across, right? If, let's say, I don't know, if the name is not catchy or so, then people, it doesn't get the attention that I think it deserves.
Yeah, if you have some weird acronym that's hard to remember. I mean.
Exactly, people just forget about it, yeah. So I think we're lucky that we actually found one that seems to be catchy, yeah. But anyway, it's not important in a way at the end of the day. What's important is that it's an eye tracking framework for fMRI experiments that allows you to do eye tracking actually without camera, and even in the data that already exists. So you can actually go back in the data that you acquired, let's say a year ago, and actually reconstruct viewing behavior from these data, and you can reanalyze those. And I think this is really important because for many reasons. One is, of course, you get an additional behavioral readout for your experiment, frankly. That's really cool. The other one is, if let's say you have two conditions that you want to compare and the viewing behavior differs between these two conditions, then also the difference in brain activity is really hard to interpret, but because maybe it's because, I don't know, let's say you have a memory paradigm, maybe the activity you see in the brain is related to memory encoding or so, or it's actually just the difference in viewing behavior. So just to interpret, to tease apart these different options, it's just important to do eye tracking. And then another one is, and there are also a couple of actually scanning artifacts associated with eye movements. And then, of course, the most important one is that a lot of people are simply interested in studying viewing behavior with fMRI and oculomotor systems. And you can use it to build better task-based models, for example. And again, this is working in existing data, so that's the cool thing here. So it, in short, it's a convolutional neural network, or it's the whole framework is centered on a convolutional neural network that takes the voxels of the eyeballs as basically like a classical regions of interest analysis, and uses these voxels to classify gaze position. And then in addition to gaze position, it gives you a predicted error score telling you how certain you can be that the decoded gaze position is correct. So this is really important. If you go back to existing data, you can now reconstruct viewing behavior, but you also get a sense of how reliable this decoding is, because let's say if you have a free movie watching data set and you don't actually have eye tracking, you don't have any test labels, you don't know what the ground truth gaze position is, the user kind of probably wants some measure of certainty for this decoding before analyzing the actual fMRI data with it. And both of this you actually get from this network. So you can, let's say, you can download a resting state data set, I don't know, 1,000 people, and reconstruct viewing behavior from these data, filter out the participants or samples that did not work, for which the decoding did not work well, and then analyze the rest, maybe accounting for viewing or for eye movement artifacts and eye movement related activity in the brain, and then just clean up your data, or actually analyze it in some other meaningful way if you want.
Yeah, I mean, it would be interesting for these, like, for example, the resting state networks, or is that the same as the default network? No, that's something else, right? Or is it?
I mean, you see the default mode network in during resting state.
Okay, right, yeah.
Often, yeah.
I mean, so, it would be interesting there just to see when people, like, with their eyes closed, or do they have their eyes closed? Anyway, just to analyze, like, how their eyes move. But yeah, I mean,
that's also actually interesting in resting state, that I think the most experiments, or a lot of experiments, use a fixation cross, so people actually have their eyes open. But a ton of other experiments just have the eyes closed, and there are a couple of studies showing that the resting state networks you uncover in these two different types of rest, if you want, are completely different, or at least they're not exactly the same, at least.
Yeah.
So that's
Which
also
makes sense.
Which makes sense, exactly. Eyes open versus eyes closed.
It is a different task.
Yeah.
Yeah.
Yeah, that's also a cool thing about deep MRI is that it works while the eyes are closed. You can actually do eye tracking while the eyes are closed, for example, during REM sleep, which just opens up also.
Sleep in the scanner?
I think they. I don't know. I've never scanned people sleeping or slept in the scanner. I think it's doable. So at least there are papers even showing that during resting state, for example, people, I think 30% of the people or so fall asleep within the first couple of minutes of the resting state, or you find at least evidence for sleep within the first 3 minutes or so in a large part of the people.
Yeah, no, actually, if I remember, like, people saying that a lot of people, not a lot, but like, they will have participants who will just fall asleep. I guess, I mean, you're lying down, it's kind of warm, and it's a kind of like monotonous noise often in the background.
Yeah, and you are in this capsule kind of protected environment. Nothing, especially during resting state, nothing really disturbs you. You're just in this, I don't know, sound box. You can't do much. You can't move either.
So one question I had, so earlier you mentioned that it doesn't work, or you can take out people for whom it didn't work, or for whom it doesn't work as well. So do you know what that depends on?
Yeah, I mean, what we do is we take the data, we pre-process it minimally, actually, do like realignment, so we correct head motion and so on in these data, and really minimal pre-processing. And then we register everything to our own template space, which is an average brain for which we know of like, I think 27 people, of which we know where they are looking. So they're fixating the screen center. So we know if the eyeballs look like this, they fixate at the screen center. And we co-register everything using nonlinear transformations to this template space. And it's a little bit of a complex co-registration procedure, but at the end of the day, it might not work exactly equally well for everybody, right? So in some people, the mask might be a little bit shifted, or they just because of anatomical reasons, they might have different distortions in the eyeballs or so. Some people just didn't do the task well or so, right? So
okay, yeah.
I don't know. Let's say if you have a viewing task, some people might just close their eyes. Actually, some people might just literally fall asleep in the scanner, and you could in theory catch those people in your data set. You could in theory account for them somehow using eye tracking.
Yeah, it seems. I mean, it almost like I remember when you presented. When you were at our institute in Hamburg, you presented, I guess, like most of your PhD, and the deep MRI was just 5 to 10 minutes or something at the end, I think.
Yeah.
It did sound like the thing which is like too good to be true, almost. Like where you have like, you start with one thing, like, hey, we can just use, you can use MRI to get eye tracking. It's like, oh, that's really cool. And it's like, even if the eyes are closed, okay, well, that makes sense. Like you don't need eyes open. And it's like, well, even for old data sets, like any fMRI data set you have, and then it just, you just stacked it more and more. It's like, wow, this.
Yeah. No, that's why we are so excited. And we also explore scanning parameters. So we scanned a subset of people with eye tracking, with camera-based eye tracking, and 9 different imaging protocols. So it's a 3x3 design for repetition time. So basically temporal resolution of your fMRI scanning sequence. And voxel size, so spatial resolution, and we have voxels from 1.5 millimeter to 2.5 millimeter, and repetition times between 800 milliseconds and 2.5 seconds, right? So we have overall, actually, the entire data set consists of 267, I believe, people, or 270, whatever.
So the training data set for the
exactly, it's a cross-validated training test data set. So all the data goes into training and into test in a cross-validated fashion. Yeah. Exactly. In a subset of people, we scan with 9 different imaging protocols, but overall we have 14. And for all of those 14 that we have, it works really well. So even for large voxels of 2.5 millimeters, or for very long TRs of 2.5 seconds, right, or repetition times, it still works quite amazingly, actually. I think one thing that should also emphasize is that in order for it to work really well, you kind of need training data for your own. I mean, if the best would be that you, let's say, if you have a data set of 30 people lying around and you want to reanalyze these data, it might make sense to scan another 5 to 10 people with an eye tracker calibration script where you know, well, people fixate at certain locations. You just present a fixation cross on the screen, use these data to train the network using the same scanning sequence. So the same distortions, same kind of similar slice package and so on. And then decode from the data that already exists. So you can't just, in theory, you could out of the box take our model weights that we will also share with the code and decode from your data set, but that's not going to be, it's not going to work extraordinarily well. It's really best if you have your own training data.
Okay, so like the best, so it's, how should we say, I mean, like one thing that sounded to me super cool about this is that, so I've never used eye tracking in or outside an MRI scanner, but a lot of people here do, and fairly regularly we see emails of people saying eye tracker's broken again or something, because they seem to be, or they couldn't calibrate it properly or whatever. It seems to be at least the one we're using here, a tool that is not as reliable as other tools we're using, let's say. And it seemed to me like super cool that you could just like get rid of the tool almost basically, but you're suggesting basically it would still be good to, just do it with a few people, and then you don't need to do it for all, at least. Yeah,
you don't really need eye tracking for that. You can scan people without an actual eye tracker, but just have fixation crosses on the screen. So you still don't need the camera.
Okay.
Yeah, so you can save these like $50,000 on the camera for the camera. And just basically scan a 5-minute calibration script for every person. So you, this is actually how it is. Yeah. So you don't really.
That's then not much of a problem then. Yeah. I mean.
Exactly. So you scan like a 5-minute scan for calibration. That's it. But of course, if you already have an eye tracker, you might just as well use it. But then, even then, in my experience, the calibration sometimes doesn't work for people. Eye tracking in the scanner is often really noisy. It depends a bit on the setup. But then for a lot of people, you will actually end up losing data, eye tracking data. And for those people, in theory, you could also, even if you have an eye tracker, you could then use deep MRI to recover the viewing behavior in those participants. Or let's say your eye tracker broke during the scan and you have 2 runs with eye tracking and 1 run without, you can just use it for the other run. Whatever. But overall, I think the big potential is really to have your own, if you have your own training data in a few participants and just decode from everybody else. And that works for existing data, but also of course for future experiments. Let's say you plan a new scanning study, you just add this 5-minute snippet of calibration script to your scanning session for every single participant, and you will have plenty of training data for your entire experiment.
So how accurate is it then? Like I'm assuming you're comparing it to standard eye tracking, or?
Yeah, it's really accurate. So we have a couple of measures, of course. So model performance, if you want, you can measure in multiple ways. I think not the best, but one way of doing it is Pearson correlation between real and predicted gaze path. And the median Pearson correlation between those two, real and predicted, is across all these people that we have, across all data sets, is at 0.9 Pearson correlation. So that's extraordinary, right?
Yeah.
And even if so Pearson correlation doesn't take into account the scaling of the data, so you could have basically co-varying signals, but let's say the scaling is completely off. Co-varying gaze positions. Let's say one is at 10-degree visual angle, the other one is at 1-degree visual angle. So in theory, you have a large Euclidean error, but the fluctuations are the same. So you will end up having a high Pearson correlation. But even if you take these scaling issues into account, you take the Euclidean error and so on, you get a really, really good model performance. So I think for many fMRI settings, I wouldn't feel comfortable putting like a number now on it because we're still working on that.
Yeah.
But it's definitely very accurate for the large majority of fMRI studies. It will be enough. So it will definitely get different where people are looking plus minus maybe 2-degree visual angle, 1-degree visual angle. It depends a little bit on your training data and so on. But I mean, the predicted error score will tell you what the predicted accuracy is given your data, your training data. So that's also pretty cool.
Yeah. So just to go to ask a question about the the precision or the resolution of the thing. So I'm just curious. You mentioned earlier that this could be cool if you have like a what's it called, like natural vision, not natural vision, but like let's say you're watching a film or something, right?
Mm-hmm.
Like let's say you want, I don't know, you're letting people watch some movie, and with people's faces or whatever, like I'm just curious, I'm wondering, like, would you be able to tell like whether people are looking at people's eyes, or I mean it probably depends, like obviously how big the thing is on the screen, but
yes, I think so. You will be able to do that. You will be able to, during the movie, tell where people are looking. And we also compare that actually during movie in a different project. I think that's also something that I presented in Hamburg when I visited you guys. It was a study on visual grid cell-like activity in the human adrenal cortex and how it progresses how it develops in development over age. And there we also decode basically eye movements from the fMRI data and compare it also directly to camera-based eye tracking during free movie watching, and also there it's really a great match. So you will, even if you overlay those two, real eye tracking and deep MRI onto the movie, you will get a really nice result.
Yeah, that's really cool.
Yeah.
I almost wish I was doing vision research now. Then I could use it. For the stuff I'm using, I don't think it's going to be much use anytime soon. But
yeah, I agree. Yeah, there's so much to do with it, I think. However, I want to emphasize that this is not only a tool for vision researchers, right? This is obviously cool for people interested in vision, but the whole viewing behavior, also the viewing behavior confound issue is present in a large part of cognitive neuroimaging, I would say. It's not limited to vision. And I personally, in my own research, I'm interested actually in the interface between perception and memory in the human brain, or how you derive like a map-like representation of the environment from your visual input. So I'm really working on this interface between more higher level memory processes and really low level vision, let's say in V1, how motion is processed, and also how this in turn affects how you remember things and where you remember things and so on. So for me, this is really a great tool because you can, not only from the vision perspective, but because I'm interested in eye movements and gaze behavior in, let's say, and signals in the hippocampus, which is not typically associated with viewing behavior, or at least currently it's not.
So do you almost think that this, I mean, you mentioned like eye movement artifacts, that kind of thing. So do you think it would make sense to make deep MRI a standard part of a pre-processing pipeline? To say like, okay, for every trial, like take out the neural activity related to eye movements?
Yeah, or account for it. So account for it or study it, right? You can, yeah, for sure. I think I mean, this is a big question, of course, that would change things quite drastically. It would basically, what you're suggesting is adding basically a seventh realignment parameter or so, like a seventh nuisance regressor to the realignment parameters. I think that's definitely possible with deep MRI, and we discussed that too. At the end of the day, of course, that depends on the user and what they want, but yeah, that's definitely possible. Yeah.
Have you found anything where you, I don't know, in either your like public data sets or your own stuff, where you, I don't know, found that like part of your results were explained by eye movement rather than some experimental thing you thought it was related
to? So in my own research, I'm doing eye tracking basically always.
Right. Okay. That's a vaguely stupid question.
But
do you know what I mean? Like, is there like, have you tried that? Or?
Yeah, we did. Yeah, so we didn't try to explain like published results and try to find like confounds in other people's stuff. That's also not the point. Absolutely not.
No, of course, but it would just show like how important it is. That's what I mean.
Yeah, exactly. But I think that we do. So we take the actually camera-based eye tracking and the predicted eye tracking data, so deep MRI derived eye tracking data, and just regress it against brain activity in a just whole brain fashion. Just see where in the brain does viewing behavior predict any activity, and we use data from Russell Epstein and Joshua Julian, so that they scanned people while they performed a visual search task. So very classical search for the L among the T, or like a distractor set task. Yeah, and we use eye tracking and deep MRI and regress it against brain activity, and we see this this gigantic brain network that is actually explained by the viewing behavior, just by basically how far the eyes moved during a TR. It's amazing. You see, of course, V1, you see parietal and frontal eye field regions, so this attention network. You see human motion process complex, but you also see retrosplenial cortex, medial parietal lobe. You also see hippocampus. You see ventral prefrontal cortex. Many regions that are not typically associated with oculomotor function. So that's I think that's what makes it so important. I think this set of analyses, it's also on the poster that we presented at VSS, really demonstrates that it is important to do eye tracking if you want to study and account for these effects, and also that deep MRI can be used to do so.
Yeah, it is really cool. Like, I'm just thinking about like my own stuff. Like I'm not doing anything that's, as I said, like directly related to vision, and it's like what people see is fairly boring. Often it's just like a few numbers on the screen or something. But you could at least, then it seems like, look where people's attention is. What are they really focusing on? Well, I guess at least using eye gaze as a proxy for attention. It sounds really cool.
Yeah, or if they are awake, if they are doing the
task. I would just check for that. Or they're just like randomly pressing something while sleeping.
Exactly.
Oh god. Yeah.
No, yeah, exactly. I think there are so many data sets and applications for it. We are excited about it, and we're glad that at least at VSS people were excited about it as well.
Yeah. And so you're using your like remaining time in Trondheim right now to get that as far as you can, or?
Yeah, deep MRI and exactly this other project I explained earlier with Ignacio Polti.
Yeah.
And I'm also planning just new stuff, of course, also for my future lab, but also with Christian Doeller, with whom I'm currently a postdoc with. I'm also planning another study, actually, another scanning study.
Okay. Yeah.
So it's basically in a conceptual state at the moment, but still, it's fun to play around with these ideas, maybe write some Psych Toolbox scripts and so on. I'm also trying to transition to Python.
Yeah. How's that going?
That's good, actually. Yeah, I mean, so far I use MATLAB, and it's, of course, the commands are different, and some of the concepts are a little bit different how you use it. But I mean, if you know how to program in one language, definitely it speeds up how you learn the other one.
Yeah, definitely. I mean, we also, we wanted to kind of transition to Python a while ago, but haven't. One of those things I think a lot of people want to do at some point, but then it's always kind of, it just slows everything down for a while, because you have to like learn all the new functions. And
yeah, exactly. That's the big problem, actually, because you always are in a way in a hurry. You also always want to get stuff done, and it's always easier in the language that you already speak or program in. Right, and then even if you try to do it in Python, but you're really comfortable in MATLAB, at some point when things are getting a bit tight or the deadline is coming out, then you switch back to the platform that you know best, and that's the problem, actually. I definitely have the same issue. So if things need to go fast, I then switch back to MATLAB, and then I spend way too much time in MATLAB again.
What is the reason for you for wanting to switch to Python? Is it like
I think it's the future, right? MATLAB. Fewer and fewer people are using MATLAB. This entire deep learning community is centered on Python. And also for me, I mean, looking forward, in a few years from now, I feel like if you're not speaking any Python, then I feel like a dinosaur somehow. I think it's what the future will hold. Yeah.
Yeah, I mean, like one thing that just annoys me about MATLAB is the license thing. Like in theory, it's very straightforward, but it's happened to me a few times where like, I don't know, you didn't have access to some toolbox somewhere or something, and then So I mean, I agree. I think it's
open access.
Exactly.
This is the main reason, and this is also the main reason why people use it that much, right? And this is amazing. It's a great reason.
Yeah, I really like. I really like the idea behind it. Just like everyone, anywhere in the world can just, you have open code, open data,
Exactly.
then
Yeah.
anyone, like, I don't know, some smart 10-year-old somewhere with an internet connection can, in theory, download your data set and analyze it in Python if he wants to.
Exactly. Yeah.
Yeah,
That's amazing. Yeah. I like that too.
and then find errors that you didn't find or something.
Yeah, I mean that's really important.
Exactly.
Yeah. Yeah.
I was wondering. So one thing that Christoph, my supervisor, once mentioned that he found, like, one thing he found a bit tricky was that you often have this carryover phase in a new position where you're doing lots of old projects,
Mm-hmm.
and that I think for him in particular, he has like, I think now he has like still like 3-4 things or something he basically wants to finish all the time, but it's difficult when you have 3 PhD students at your feet. It could be, you have other stuff to do. So I was curious, like, is in your new position, whenever it will start, is that closely related to what you're doing now, or is there kind of a bit of a step? I mean, it sounds like it would be pretty similar.
Yeah. Maybe just a short recap of what I've been working on.
Yeah.
I started basically in a vision lab and in a neuroimaging lab with Andreas Bartels, and worked on how visual cortices integrate visual and motor information, eye movement information, in a visual motion task. So I was wondering how the visual system stabilizes perception during self-motion. That's basically how I entered this entire neuroimaging field. And back then I remembered, or I remember thinking that knowing how the visual system might be able to stabilize perception, I felt like I'm really understanding a piece of the puzzle, how we just experience of our entire visual experience, how we experience the world and so on. But then in the course of that study, I read a lot about place cells, grid cells, head direction cells, so cells that you would find in the hippocampal formation, medial temporal lobe in humans. And I always felt they are really related to what I was working on, because they basically derive a stable representation of the world from these visual inputs that I've been working on. But at the same time, most of these data were acquired in rodents, navigating in boxes, and it was electrophysiological recordings. And I was working on visual motion and fMRI, and at the same time, simultaneously I felt somehow related to this work, but at the same time very distant, right?
Yeah.
Also methodologically. And that changed when I read a paper by Beth Buffalo from Seattle, who recorded in the entorhinal cortex of monkeys. And they showed that these grid cells, and just I'll come back to what grid cells actually are, but they recorded grid cells in a monkey just looking at images. So suddenly these coding principles in the hippocampal formation were just much closer to what I was working on, which is they played out in visual space, which is what I felt comfortable working with. And then another paper by my current PI, Christian Doeller, who showed that these grid cell-like signals, you can pick them up in fMRI while people navigate in virtual environments, or at least something that we think is a proxy of grid cell activity. And these two papers combined really sealed the deal for me. I noticed, wow, I can actually work with the tools I already have, with the skills I already have, I can actually work on this topic as well and have a bigger perspective on how visual cortex and these hippocampal cortices somehow how they interface. So we ran a study with Christian, and then also a couple of follow-up studies. And this is the core of my PhD, if you want, along with the paper that came out 2 weeks ago in Nature Communications, where we show head direction modulation in fMRI as well, using an encoding model. And overall, my PhD, I felt, was really focused on this forward sweep of information from visual cortex to the hippocampus, how you derive a stable representation. But now looking forward into my postdoc, I feel I actually want to go the other route. I want to know what these higher order visual spatial coding principles actually allow you to do. And I think This is there's two things. One is they help to integrate visual experiences into memory and retrieve them when you need them, and they allow you to plan your behavior in space. And in particular, in a TICS review published in 2018 with Josh Julian, I proposed that the human hippocampal formation represents this coordinate system of visual space that allows you to do these things, memory recall and formation and planning of viewing behavior in this case. And this is what I'm gonna study in my postdoc as well.
So maybe could you briefly describe your Nature Neuroscience paper?
Mm-hmm. Yeah, so the entire paper builds on the idea that there are grid cells in the brain, and they were discovered in rodents by the Moser lab, who is basically the core of the institute I'm working at now, at the Kavli Institute for Systems Neuroscience in Trondheim, Norway. And they discovered these cells that represent the environment with a grid-like code. So these cells, if you record the activity of a single cell, it would fire at distinct locations in the different locations in the room while the rodent is running around. And these locations are not just randomly distributed, they actually tile up the environment in a hexagonal grid-like fashion. So it's amazing. For me, this is definitely among the most beautiful discoveries in neuroscience, really. I mean, the pattern is absolutely striking. And I was just hooked by it when seeing this. I thought, why is that? This is insane, right? And I think a lot of people felt that way when they saw these grid cells. And also the discovery was rewarded with a Nobel Prize, together with John O'Keefe, who discovered a related cell type called the place cell, which fires only at one location in the room. And those cells can be found in the adjacent hippocampus. But I was focusing on these grid cells, and I tried to find evidence for these grid cells that also Beth Buffalo found in monkeys as a function of where people are, where the non-human primate, the macaque, was looking on the screen. They found that a similar grid-like pattern emerged in this viewing task. And I was wondering, can we find evidence for these visual grid cell-like codes also in the human entorhinal cortex? So to study that, I went back to what I knew already from Andreas Bartel's lab, which is this visual tracking task. So back then I used a smooth pursuit task where people fixate at a fixation target that's just smoothly moving across the screen.
Like so
yeah, so in my case, it was always a very clear predefined trajectory that allowed you to sample all directions equally with like constant speed and all these factors matched.
But like the participants didn't know when it was going to change or anything like that, or
well, I mean, I think after a while it does become a little bit predictable, I guess. But the actual task they performed was memorizing locations on the screen. So they fixated with their eyes these moving fixation targets, but the actual task that we got a behavioral readout from was memorizing locations on the screen that just popped up. There was like targets popping up on the screen, and they would memorize the location of these targets on the screen and report them in the course of several trials. So they basically formed, during this smooth pursuit task, they formed a spatial memory representation of the visual field and where these locations are. So that was the task. And then we analyzed the entorhinal fMRI signal as a function of eye movement direction. And now you would think, why would you, I just told you that grid cells encode the location of the animal in space. Why are we now looking at direction of eye movements? It's quite a stretch, right? So for that, you need to understand that this grid pattern that these cells express is built up from hexagonal patches. And if you rotate this entire pattern, this firing rate map, if you rotate it, it would look very similar every 60 degrees of rotation. So it's modulated by 60-degree or 6-fold rotationally symmetric. And the other thing is that if you walk on the main axes of this grid, you would cross over more firing fields than if you walk off or orthogonal of these axes. So overall, the prediction was that if there are a lot of grid cells in the brain, in these fMRI voxels, then you should see a 6-fold rotationally symmetric signal as a function of movement direction as well, because the location signal, for various reasons that are too complex here now, but the positional signal we didn't expect to see in the fMRI signal, but the directional signal should come out, and that's what we looked for. So we looked specifically for the six-fold rotational symmetry in eye movement direction, and that's exactly what we observed in this task, in this viewing task. So there was no, I mean, think about it. In rodents, these cells were discovered while the rodent was running around in a box, and now we can measure these things in fMRI while people look at the screen. So that's pretty, I thought this was pretty cool. And most beautifully, it was also published back to back with a completely independent paper from a different group, from Russell Epstein's group, and led by Josh Julian, who later became a postdoc in our group, and we also collaborated on various projects. So that was also really cool. But these two projects were completely independent, and they made exactly the same observations. So giving you a lot of confidence, also putting it out. That was, for me, also just a beautiful feeling. I mean, in the beginning, of course, I think Josh and I both were a little bit like, holy shit, somebody is doing the same thing as me. It's just like it is for a young PhD student, knowing, I mean, you get a heart attack when you know that somebody else is doing remotely the same as you. some other person.
Brilliant idea.
No, but I mean, that might be your brilliant career, right?
Yeah, of course.
Who knows? Anyway, or just, I mean, you spend so much thought and so much effort into a project, especially in the beginning. It takes you so much dedication to get a project going and finishing it. And then, of course, you get a heart attack if anybody remotely looks at the same fMRI machine as you. Then you think like, oh, holy cow, they're doing the same.
Yeah.
So in the beginning, it was.
Find out that they were doing the same thing. Was it just when you submitted it to the journal, or was it before?
No, we found out before, actually, at a conference, at SFN.
Okay. So did you like coordinate this and say let's submit it together, or was it? I'm always curious, like, because there's been a few papers I've seen recently, like the development of the grid cell and place cell development. That was also a back-to-back publication by, I think, John O'Keefe's group and the Moses group. And I always wonder, like, whether that's like partly intentional or just complete coincidence.
I mean, much of this is probably agreed upon beforehand, and I think I'm actually a big fan of that. Just from the get-go, speak as transparently as possible about what you're doing. I think nobody tries to step on your feet, or at least very few people really try to screw you over. Or then I think most people, if you are transparent, will also be transparent.
Yeah.
And in case of Russ and Josh, we just talked at the conference the first time, actually, at SFN. And then back then they were really far ahead of us, I think, in this project, and they couldn't wait for us, basically. But then during the submission process, so they submitted, and the paper was sent to us for review. So we were invited to review it.
Yeah.
Or Christian was invited to review it. But then we said we cannot possibly review this paper. We're obviously not naive, or we have a conflict of interest.
Yeah.
Exactly. So we responded to the editor. We're really sorry. This looks like great work, but we cannot review this. But we have very similar results that we also would like to submit very soon. So is it okay if we submit? And then he invited us to submit it as well. And then during the review process, these papers actually synced up and came out together.
Okay.
Yeah. So it worked out beautifully in the end. But I mean, in the beginning, of course, everybody, I think, it was just a lack of communication. It was not really miscommunication or so, or nobody tried to screw anybody over, but it was just a lack of communication in the beginning between the two of us, both groups. And in the end, it worked out, and this was the most beautiful thing of all, I think, for me.
Yeah, it must, like, from the outside, it seems like it must be amazing to have like a really cool paper immediately, independently kind of, what's the word? Replicated or confirmed.
Yeah, replicated each other, basically. Yeah. Exactly. Yeah, and I mean, the signal itself, this six-fold rotational symmetry, was then also replicated by Tobias Staudigl, who is now a PI in Munich, in MEG, actually.
Yeah, MEG.
So he also looked at in very similar viewing tasks. He looked at
entorhinal cortex.
Yeah, I mean, it's source reconstructed to the medial temporal lobe. You cannot really say it's like this is entorhinal cortex, but it's the medial temporal lobe. Really the signal is coming from where you would expect it to come from. also in MEG.
Okay, that's cool.
Yeah. And now we also replicated it in yet another data set that I presented in Hamburg.
Which one was that?
In this aging or in this developmental data set. Yeah.
Can we talk about that briefly?
Yeah, sure. Yeah. So we downloaded a publicly available data set of people between 5 and I think 20 years old, 21 overall. I think for the oldest group of people, we don't have too many subjects. So we kind of cut off the oldest group. But yeah, it's a really range of it's a nice age range in the participant group, and we run these grid cell-like analyses. This is what we call the hexadirectional analysis because it's six-fold rotational symmetric. This hexadirectional analysis we ran on all of these people in a viewing task. It's a movie watching, so I think people watch Finding Nemo. No, Despicable Me, of course. Finding Nemo is yet a different data set. Sorry.
This is a crucial difference.
Exactly, crucial difference. Yeah, both great movies. Exactly. So Despicable Me, they watch like a short snippet, and we see that this grid cell-like signal actually builds up in the course of development. So it's we see that the amplitude of the signal, it's a bit complex, but the amplitude of the overall signal, this modulation in the fMRI signal, is more or less the same in all. But the distinct, basically, where these signals peak or which directions have a high activity and which directions have a low activity, this is more stable in the old than in the young. So in the young, it's as if these visual grid cells, if you want, or visual grid cell-like signals are not anchored to the screen as much as in the adults, even though the overall modulation of the signal is similar. It's just more stable in the adults. So we interpret it as the cognitive map being more anchored to external cues in the adults versus the children, because it's relative to the movie. It's these signal modulations become more stable, so they are aligned to the screen, if you want.
And Chris, if I remember correctly, in the rodent literature about the developmental stuff, as in like how the place cell, grid cell, border cells, these things, how they develop in rodents. If I remember correctly, it seems like the rodents, they kind of start out with proximity to borders, so the border cells are very strong, and then I can't remember like the exact order of them, but it seems like basically you have like some sort of, you look for external cues first, like distance to walls and that kind of thing, and then as they get a bit older, they develop this map where they can move kind of free, like kind of more independently of these landmarks.
Yeah.
Can you look for that at all or is it, I don't know whether you can like look, for example, look for boundary vector cells or something like that, or is that just not, we don't have the analysis for that?
We discussed it a lot actually, and we even ran some boundary analyses, but they didn't really show anything. I mean, one thing we did is just we tiled up the entire screen in different bins and just compared boundary bins versus center bins, but you don't really see a difference between those two. At least so far, we don't see anything interesting popping up. And it's always, I mean, in fMRI, it's always complicated to think about where. I think the beauty of this kind of research, or at least what brought me into this fMRI research, this specific question, for example, was that you have a very clear prediction taken from electrophysiology, and you test it. You think about how would this translate to a population level signal. Wow, then you have a prediction that you can now test in fMRI. And this nice taking an hypothesis from EFIS and testing it on population level in fMRI, what was so attractive for me in a way in this project. That's what I thought was really beautiful in these previous studies, also from Christian, for example. It was this beautiful single cell inspired finding that he put forward. And for boundary vector cells, or boundary cells, or border cells, at least to me, it's not very clear how the population level prediction would look like. Maybe just comparing boundary versus center bins is actually not the best thing to do. I think in these, in Beth Buffalo's data, at least, they see that there's something like a visual border cell that does fire along the boundaries of the visual display, but not at boundaries within that display. So of course, the movie itself that they watch, for example, or the image itself might have some different edges and stuff inside the picture. and these cells do not fire at those edges. At least what they in the data that I've seen, these cells do not fire at these edges within the image, but at the edge of the image. And that's what we looked for, but we didn't see it. Yeah. But maybe our prediction is just wrong. Who knows?
Yeah. But I mean, as you said, like it is a kind of a big step to go from single cell recordings in rodents to population-based analyses in fMRI in humans. I mean, Christian Doeller, it was a Nature paper, right? From being able to show that it's possible with grid cells, right? Like that by itself is a major achievement.
You need to, of course, emphasize that also I don't want to sound like I'm suggesting that fMRI measures grid cells. This is not what I'm saying. I'm just saying we took a prediction from EFIS literature and tested it on fMRI, and it was confirmed. What the actual link is between these two worlds, I have no idea about, right? There might be, it might end up actually that none of the fMRI signal is explained by like grid cell spikes in itself, but maybe it's the inputs to grid cells that might actually be not perfectly correlated with the spiking of a grid cell. It might also be it's actually some head direction cell signal that somehow is modulated by 6 degrees. There's some evidence also for also in our group, and Tobias Navarro-Schröder is analyzing LFP data, seeing and also in MEG and recording in other people's recordings data, you see this 6-fold symmetry also in the LFP. And what really drives it, whether it's spiking activity of grid cells, is still kind of unknown. We like to believe that, but that link is not fully closed, I would say.
Yeah, thank you.
And
That's a good point.
don't interpret
I immediately went to it's grid cells. But yeah, you're right.
Of course, I mean it's hard. At least I don't know any other explanation for these results. What other stuff than grid cells would lead to a 6-fold rotational symmetry? I don't know. So, and the prediction comes from grid cells, and it was confirmed. That's where we are at now.
But I guess the more important point, isn't it, also that like it's more about the computational mechanisms behind it rather than whether a single cell does it or networks of cells. I think to some extent, to me, that's maybe not even the most interesting thing, but the thing that you can see like the same computational patterns is, I think, to me, more interesting anyway.
Yeah, for me, it always, when I think about fMRI and my own data, I always think about, especially for the computation, if you want to discuss that level. I mean, fMRI is sensitive to synaptic processing, which usually reflects the inputs to the system, and of course local processing, right? Just wherever there is transmitter reuptake, you have a lot of metabolic activity somehow, and you might find an fMRI signal or a change in fMRI signal. But does that really mean that there is a computation going on? If let's say if there are inputs coming into the system, let's assume there's a cell somewhere downstream, there's a receiver cell, and it receives some synaptic inputs, but none of these inputs is enough to actually make that cell spike. Then you would still have potentially an fMRI signal increase, but in that receiver cell, potentially there's not a single spike. So would you call that a computation then? Or you could say, well, there's synaptic computations, but what are they good for if the receiver neuron is not spiking? So I always think about finding these results finding fMRI effects does not necessarily mean that the computation is going on in that area where you find it in. At least there is a chance that it might actually origin somewhere else.
Yeah, that's a very good point.
How do you see that?
To be honest, I haven't really thought about fMRI that much in detail in a sense, because I mean most of my stuff has been in, I mean the two master's projects I did do were with MEG and EEG, and only now I've done behavioral stuff so far since starting my PhD. Mm-hmm. So this is kind of something that's still ahead of me, something I've been saying I'll start soon for like a year now. But to be honest, I don't think I have a particularly qualified point on that. Like, for example, I don't exactly, like the other thing is, like the specifics of where the bold signal comes from. Mm-hmm. I don't really know that much about it, to be honest. yeah, I'm not even sure how much is known about it.
I mean, I think there are plenty of studies on it, but there are still many unknowns, I would say. And of course, let's say spiking let's say the receiver neuron does spike, then the chance of neighboring synapses picking up that spike is also very high. So of course, let's say if there is activity in a region, let's say spiking, even output activity, the chance of this spike receiving neighboring neurons is also fairly high. So this intrinsic connectivity is usually very strong in many regions. So you might end up seeing it in the same, you might see the fMRI signal in the area where the spiking happens. All I'm saying is you cannot really make that causal link, I think, in the data. And just being aware of that, I think, is important. At least for me, I try to remind myself always.
Yeah, right. I'm really glad you
fact.
did. Yeah. I mean, it's just so one question here. So you said you started off in vision itself, right? Or you studied biology even at the beginning, right? In your diploma or bachelor's or whatever it was. I'm curious, why do vision in humans if you can do it in animals and have, much more precise knowledge about what you're actually measuring? So like what made you interested in studying vision in humans specifically rather than doing it in animals?
Well, ultimately for me, most of the animal research is, I think it depends a bit on what your motivation is to do science at all. For me, I came into science also for personal reasons, because I just wanted to know what this, I mean, it sounds a little bit, I don't know, dramatic, but I mean, I came into this business because I was curious. I wanted to know how this world works and who I am and how I perceive stuff. And studying biology, I simply thought, well, I could now study, I don't know, some formation of trees or some specific animal or so. And then at the end of the day, I would go home and know a bit more about this tree, how it works.
Yeah.
Would be amazing. I'm not saying this is really important science, and I love reading about this and so on. But I felt, for me personally, it was not as satisfying as knowing about how I perceive all of those things. Let's say understanding how I perceive color made me go home at the end of the day of university thinking, wow, I learned now something, how I see this tree, this animal, this car, basically how I interface with reality, or what I can interface with, at least. So it's more, for me at least, it was more like a philosophical, I think, motivation to get into science, and it's basically a self-discovery in the beginning. And from that point of view, I just wanted to study vision in humans, because ultimately that's who I am, right? And who all of us are. And it's also, I think, the methodology actually also I liked a lot. So I like the methods also. I like fMRI, I like neuroimaging, I like having whole brain resolution. A lot of questions you can simply ask only on network level. I mean, now, of course, with calcium imaging and neuropixel probes and so on, you can ask a lot of these questions also in animals. But when I started off a couple of years ago, of course, people did calcium imaging back then, but it was not the thing that it is now. It was still, you studied a little patch of cortex, and oftentimes you studied, let's say, fewer than 50 cells, let's say, right?
Yeah.
In most of these recordings, or in a lot of them at least. Yeah, I just wanted to go for humans and have a bigger perspective on things, on the whole brain, how different areas interact. And I think this question that I'm in a way posing also is in my PhD was, how do you derive a stable representation of the environment from visual inputs? That you cannot, that entire question you cannot address by looking at V1 in isolation, or hippocampus in isolation, or retrosplenial cortex in isolation. You need to understand the entire pathway, and that's, for me at least, a convenient way of doing that is just a tool that allows you to give access to, that gives you access to all of these things simultaneously.
I was about to say, it sounds like that is exactly what you're doing, right? You're looking at how, let's say, more visual areas and hippocampus, for example, interact. I guess you said, like, in your PhD, you looked from V1 to hippocampus, and now you want to look at the other way, basically. So yeah, I see how
It's a network, right? For me, in my understanding, the entire brain is a network. No single area evolved in isolation. Also, the function of an area can only be understood in the context of what all other areas are doing. It's not, you cannot isolate hippocampus and understand what it is doing. You cannot cut off neocortex. Of course, you can learn a lot about the hippocampus doing that, right? You can, of course, understand its anatomy, intrinsic connectivity, maybe its cell types, all these sorts of stuff. But ultimately, all the areas are embedded in a network that comprises all areas, and they are functioning only. They, in my understanding at least, they function the way they do because this entire network is there. And yeah, I find this network level perspective is necessary, and it's, at least fMRI does give you access to that. Of course, there's a lot of criticism about fMRI also because it's imprecise, it's not clear what it actually measures. Temporal resolution is often poor, even though people like Nico Schuck, for example, put forward that actually the resolution might be much higher than people thought so far, temporal resolution. But yeah, it gives you this global perspective on the brain, and it measures really everything that goes on in a voxel. Let's say there is a spike, it will also trigger a lot of other spiking. It will also trigger glia cells, for example, that reuptake the transmitters and all this sorts of stuff. So it measures at least everything that goes on in that voxel, not, let's say, these few % of the metabolic demand that's associated with the actual spike. So you, in a way, yeah, get...
Is that a good thing if you're measuring everything? I mean, doesn't that just, if you're measuring stuff you're not interested in, couldn't that just increase your decrease your signal to noise ratio?
I don't think it's really it's good. It's neither good nor bad, I would say. It's just important that you know about it, right? You just need to know what you're measuring. It's not inherently bad or good to measure everything that goes on in a voxel. And then, of course, there are all sorts of other problems, multiple comparisons and all sorts of stuff, which need to be addressed, of course, and are being addressed, and this is really important. But you need to understand, you need to be aware of what you're measuring. That's just all what I'm saying. And it's neither good or bad. It's a signal that you get. It tells you something about what the brain does. Yes, it might not be individual spiking neurons. It's still, for me, very interesting. And I truly believe that it does measure indirectly neuronal activity. I mean, look at, for example, retinotopy in V1, for example. I mean, it's such an amazing, strong signal that you get in V1, how visual stimuli are represented in the brain. You can actually draw letters into the cortex using different stimuli. You can, let's say, if you present an M on the screen, you will see that M reflected on the cortical surface. I mean, how would that even be possible if it doesn't measure anything meaningful? And I mean, if, let's say, the fact that there are a lot of glia cells in there, I'm not an expert on that, really not, but maybe it even helps you pick up the signal because it amplifies the signal, if you want. Because let's say there's one neuron spiking, then a lot of cells would reuptake the transmitter and overall potentially boost the metabolic demand in that voxel. So in a way, it could actually, for fMRI, be beneficial that there's a lot of stuff happening because it will amplify the neuronal signal. Of course, it will add noise as well. But yeah, it's...
Yeah, and I don't know much about glia cells either, or... I mean, I had one small module on neuron glia interactions, and I mean, the big picture message I remember from that is that, glia cells are involved in computations and all these kind of things.
Mm-hmm.
They take up calcium or whatever, they release it, at least in some situations. So, I mean, yeah, you would assume that there's a decent chance it has something to do with the neuron activity also.
Yeah, I think so too. Yeah. I mean, it might not be in all of the cases, but in some. And I think there's beautiful evidence that it does so in some. And yeah, visual cortex, that's another thing actually in the beginning, because you asked me why I studied vision in humans, why not in animals. Just seeing retinotopy in humans, and something called population receptive field mapping just blew my mind as an undergrad. Just seeing basically in your own data, in your own brain. I was also a frequent participant in studies back in Tübingen where I studied, of course. Basically, that's how I financed my bachelor's and my master's, if you want. Also, of course, my parents supported me as well, but also in addition, I got some money on the side from just participating in every single project there is, and just seeing basically my own brain activity in these data, as in response to these different tasks that I was performing.
Was that
beautiful for me.
In Andreas Bartel's group, or was that
in all sorts of groups? Yeah, but also in the group. Back then I was basically the pilot subject for most of the studies.
That's pretty good, going from participant to. You did your master's project there, or what was it?
I did, yeah. My master's, and then I worked as a research assistant in the lab as well for quite a while.
So I'm not particularly familiar with the biology curriculum, but I'm somewhat surprised that you'd have like fMRI retinotopic images in undergraduate biology. Was that like as part of a, I don't know, vision module, or how did you come across those first?
Actually, I think in the actual university studies, the only fMRI study we discussed was the dead salmon, which you might be aware of.
Of course.
So I'm not sure if everybody at the biology department was a big fan of fMRI. Yeah, but fMRI was not a popular topic overall, I think I felt at university. But I had friends who studied psychology also, and we just got into contact with the Max Planck Institute there. I think I actively reached out to human researchers who then were much closer to fMRI and this kind of thing. So in the actual studies, I think it gave me a good understanding of the neurophysiology, and overall the physiology is what reeled me in, if you want, right? Understanding how stuff maps onto neurons, and even how the kidney works. So this kind of, you understand how you work, and by doing so you understand, or at least I feel I understand what I can even know about the world. If you want.
Do you think that's so I'm always like, one thing I always, or not always, but I occasionally think about is like what or sometimes when like even some people ask me who maybe are doing a bachelor's project or something like that, or like in their bachelor's what they should do for their master's. So I'm always curious like what path in a sense people should take if they want to do, let's say, something like what you're doing. So I'm curious, so was your bachelor's was in biology, and then your master's also, or was that in, what was the topic there officially?
Neurobiology.
So I'm just, again, like a very like cellular kind of course, or was that also
cellular, and I would say behavioral also. So I had an amazing course on primate biology and behavior with Andreas Nau in Tübingen. That was amazing. Also, I remember I gave a talk on vocalization behavior. Just basically, something like a rudimentary language, if you want. I would say it's mostly cellular and behavioral studies that we discussed, probably.
Okay, so I'm curious, like, what do you think is the advantage of having that background? Like, is it something like you said earlier, like you know what you can, that you're maybe more aware of maybe asking what you can find out with certain signals, or what you can even know with these kind of things? Or what do you think is maybe the advantage of having studied biology versus psychology, mathematics, physics, computer science, whatever?
It's a difficult question because I didn't study all these other things. But for me, my entire thinking is inspired by my knowledge about neurons and single cells and electrophysiology and just biology overall. This is where I come from. And at the end of the day, I use fMRI to study the brain, and the brain doesn't compute in voxels, right? It also doesn't compute in signal to noise ratios, or it might do, but not on fMRI scale. At the end of the day, what I'm interested in is the biology, right? And the cognition that it gives rise to. And for that, at least my thinking is based on my biological background, and I wouldn't want to miss it. Also, I would study biology again, maybe actually more physics as well. If I could just start over again, I would probably study like a combination of general physics. So the two approaches to reality. So basically studying how the world out there likely is, and then also how I perceive it, knowing about my own sensors, knowing about how I interface, knowing about how I acquire knowledge about the world. But in a way, these two things go hand in hand for me. If I want to know what's there out there, I kind of need to understand how I measure stuff, how my body is measuring stuff, how I work, how my brain works. And I feel, at least, I mean, privately, I like to read about, I don't know, physics, also astrophysics and so on. I'm always mind blown about what they are able to show. Maybe I also don't understand it enough to know what's wrong with it. But I mean, if people, I don't know, if I actually I do read some papers even sometimes on just some off-topic stuff, at least for my own research, irrelevant stuff. And if I read a paper about how different atmospheres are measured on distant planets, and I feel like this is insane. And I would love to, at least in my studies, I would have loved to have more of that, I think.
Do you think that kind of stuff is... I always wonder whether that stuff is easily teachable, or, like, because then it's a module you have to take and then they tell you certain things, whereas if it's kind of something you just explore on your own, I feel like often that can maybe not enhance the sense of wonder more, but I don't know, maybe it depends on like what kind of student you also are.
I didn't quite get that. What do you mean exactly?
Well, so it sounded to me as if you meant that it would be cool if in your bachelor's or master's you would have had maybe a module on, well, I'm not entirely sure what, was it just on like a different topic, like something that's like completely left field and has nothing to do with biology? Or what exactly would you have liked to have had?
Basically a full study in physics.
Okay.
If you want.
Okay. You just want to do physics. Okay.
In addition to a full study in biology, yeah.
Okay. I see. Okay.
Actually, if you think of it, also knowing about the fMRI signal, you actually learn a lot about physics, or you need to know a bit about physics. So in a way, I feel the research I am doing, and also you are doing, is really at the interface of many of these disciplines, right? You take inspiration from maybe biology, from ePhys or so, but it only matters to you because you want to understand maybe the cognition behind it, or basically from a psychological point of view, what this means to you, all this biology. But to do so, you need to know how your measuring device works, if you want, what it measures. At least in fMRI, you need a decent understanding of physics if you want to do it right. If you develop your own sequences and so on, you are basically forced to dig into more of the details, which is good, I think. You also need, from a statistical point of view, need a lot of or a strong background, basically. You need to understand all these tests that you're doing, all these data cleanup, signal processing. You need to understand programming, so there's some computer science coming in as well. I feel like it's not one or the other. You need this bigger perspective and interdisciplinary, all these interactions. And I was always lucky that I've been working with people from very diverse backgrounds. For example, psychologists. My office mate here, Markus Frey, who co-developed Deep MRI with me. He's a computer scientist by training. So machine learning and AI, basically, that's where he comes from. I'm a biologist using fMRI and eye tracking. and the two of us in the same room is amazing, because we try to, or we address the same problems, but from a completely different perspective, right? And the fact that we both have a background in coding at least, or some programming skills at least, gives us a language that we can talk in. And I think we both have a decent understanding of signal processing, right? All these basics, how I don't know, how does a filter work? What is convolution? All of these things. If you know about this stuff and a bit of statistics, you can at least interface with these other disciplines a bit easier, I feel. At least for me, this was the most very fruitful interaction with Markus, and overall with computer scientists. And then psychologists, of course. There are a lot. Christian Doeller, my PI, is a psychologist by training. I supervised a master student who actually pursued a physics study here in Trondheim. Also, again, basically coming from a different perspective. Yeah.
So what was he doing with physics? Like, was it a MRI physics thing, or what was his?
Yeah, we analyzed some data. Yeah, so data analysis project. So 70 data. But I think for him it was also interesting because at the end of the day, this is the analysis part, the analytical part of the project, and identifying the problem, trying to solve it, writing code to solve it, interpreting the results, visualizing it, all this sorts of stuff is basically the same no matter what you do, what kind of field you are in. And as I said, I mean, fMRI is a physics-heavy field, if you want. And for him, I think that gave him a natural entryway also. So he came from physics and wanted to know a bit more about the brain, but entered the field via this, how does fMRI work, kind of, this was his approach to the field, I think. And then he learned more and more about the brain as well. And I went the other way, right? I came from, this is a cell, the mitochondria are the powerhouse of the cell. So
it's a classic.
That's a classic. To, how does a scanner work? I don't know, what's statistics? What is programming?
Yeah. I'm curious about, like, one thing I always find a bit difficult is because it's so interdisciplinary, and you can go like, you know, it seems like in any direction you want to go, there's just so much stuff you can look at. Like, even if you want to, I mean, if you just do a very simple experiment with very simple statistics, you know, just in the statistics you can, you know, I mean, there's people doing PhDs in statistics, right? So I'm curious, how do you decide how much to look into a field, like how much you need to know for a field? Let's say for MRI physics, like there's a point where you have to stop looking into how the physics of it works, otherwise you're doing physics. So I'm curious, how do you kind of handle this? Well, at least what for me is quite difficult to figure out when I've like learned something at enough detail and when I should move on with my main stuff.
Mm. Yeah, that's a very good question. I don't think I have a very good answer for it. I would think that when you know enough about some basically side topic.
Yeah. I mean, I think I might sometimes go a bit too far on some things where you realize, like, okay, reading one paper probably would have been enough. Like, you did need to go like 2-3 citations into that paper, like references into that paper. But I don't know whether it's just something you have to like learn by experience and see, like, okay, I don't know, the last few times I learned lots of stuff that is just, I just can't use in this, or yeah. But then I always find like sometimes you also maybe, you know, you're missing out on some sort of serendipitous finding because you just don't understand the thing well enough.
At the end of the day, I think it's probably a question of practicalities. You need to move forward with your project, right? At some point, you're just restricted by time, limited by time. In theory, I would love to know everything about everything. That's clearly not possible. And I don't know. I don't think I don't feel I lose myself too much in some side topics. Maybe I do, but I don't experience it that way. At some point, it just becomes too detailed for me, or I think this is not relevant for my own work. I think you might. I think one good advice could be to think about how this actually informs what you are doing in your own work, and if you feel like this is actually, or maybe or how it speaks to some private interest of yours. But at some point, you'll notice, well, this is actually. If you ask yourself, what I'm just reading, does this get me anywhere in my private search for knowledge or in my work? And if the answer is not really, then you might have gone too far down the rabbit hole.
Yeah.
Not sure.
But yeah. So I like one example that I'm curious, like how much, for example, I think this is a question that many people, especially those who come from psychology, ask themselves, like how much math do you need to know? I think knowing none is probably not a good idea. And doing an entire maths degree is maybe a bit much, but like, so how have you, I don't know, did you had any in your undergraduate, bachelor's, masters, or did you have to like learn all of that yourself, or how did you go about that?
Yeah, so I had some of course, in bachelor's and masters. That was also very useful. I wouldn't call myself like a math expert or so. I think I know the stuff. For me, at least, I think it's again a practicality issue. I don't really like copying code from other people, so I try to implement it myself usually. Sometimes I get a script from somebody, but I would never use it. I would actually then look at how it works and then implement my own script based on it. And to implement it yourself, you kind of need to dig into the math, but then you're already limited to what is relevant to you.
You mean because you have like an outline of what the other person did? Or?
I mean, you have an objective, and you kind of search for the maths that get you there, And I don't know. I mean, overall, I feel like I would love to have a much better or more solid understanding of math than I do. But I feel like I do have enough to pursue my work. And in the end, at the end of the day, a lot of the stuff that you would learn, let's say in pursuing a maths degree, is not directly relevant to your practical work as a biologist. It can inform it. I think if you have a solid background in math, it's amazing. Your starting position is a completely different one. But it's not absolutely necessary, and you will develop the skills that you need to pursue your work along the way if you're dedicated. And at the end of the day, it's all a question of dedication for me, or maybe if you want to bring in this word commitment, for me it's all about commitment. You need to be committed to what you're doing, and if you are committed to neuroscience, then you're automatically committed to learning the skills you need, and that includes math, some basic knowledge of physics, some psychology, biology. You need to, I think this is the way forward for us overall as a discipline. Maybe that's a big question now, but overall as a discipline, it's the way forward is to grow in all of these directions and have people that at the interface of these disciplines and make them talk.
Do you see yourself then as like a person at the interface, like someone connecting people from other areas, or someone who is like very heavily based on biology and fMRI, whatever, and then you're kind of, do you see where I'm trying to get at? Like, I feel like there's some people who are maybe really good at communicating with lots of different kinds of people and bringing them together, and others maybe who are the other kinds of people who then say, okay, well, let's say, for example, you come from a biology background and you say, okay, I just want to, now I'm collaborating with, what's his name, Markus Frey, a computer scientist. Is that kind of how you see your role in this, or?
I mean, it might go too far, I don't know. I'm just a guy like everybody else, and I'm doing what I love. I think I do bring people together, but I'm also brought to other people by other people. It's an interaction. I don't know. definitely I do bring people together, and I'm really grateful for a lot of people in my surroundings, in my environment that connect me to other people. So I guess the easy answer is yes, but so are a lot of other people, and I think this is again, this is a network issue. It's like the brain, right? Everybody co-evolved in a big gigantic network, and yeah, everybody is a connector if you want.
I have, if I can change topic a bit, I have one question I'm curious about, and this is a very dumb topic almost, but it's the use of Twitter. Because I think over the last, I don't know, since whenever it was started, I think over the last 5-6 years or something, I've occasionally started a Twitter account and then never knew what to do with it, and then basically deleted it. And like 3 years later, I was like, I should maybe try and do Twitter again. And it seems to me as if you're quite, I mean, it's not like you're tweeting all the time, but it seems to me you have quite a few followers for a scientist on Twitter, right? If I remember correctly. And I find like you looking at your tweets is pretty informative.
So thank you.
So I'm curious, like, how do you, like, what do you put on there? How do you decide what to put on there, what not to put on there?
I mean, I try to limit it to science. I don't really engage with political discussions on Facebook, on Twitter or Facebook. I'm not active on Facebook at all, actually. But on Twitter, I am active indeed. No politics. I try to stay with science and evidence and papers. I think it's important to do science communication, which is one of the reasons I'm doing that. The other one is just I love to share stuff that I like. That goes for science, but that also goes for music. I don't post music on Twitter, but I do spam my friends all the time with, like, look at this YouTube video. This is amazing. This is a great song. So I would also send these recommendations to people that do not ask for it necessarily.
Thanks for that, Matthias.
Yeah, exactly. Thanks for that song. But I mean, on Twitter, at least, the people who follow you, they could always unfollow you, right? Yeah. So these are the two reasons. I think science communication is important, and I love sharing stuff I love.
So it's like two fellow scientists, and is it also then like for the general, I mean, whoever the general audience is, like science communication?
Yeah, so I did, for example, for Real Scientist. That's also a Twitter account or a website.
So what do they do?
Yeah, so they share. So that's different people host that Twitter account for, let's say, a week, and then they tweet about their work, daily stuff, how the work is, kind of about the practicalities of the work. This is my desk. This is my.
Oh, sorry, like literally.
This is my lunchroom.
I see.
Exactly. Okay. But then also, so your workplace, but also about your science, or that actually might be anything. There could be people that work in industry, for example, or do academic research. So it's a broad scope. I was one of the hosts, speaking to the German public about science and tried to explain to them what grid cells are and place cells and memory formation, episodic memories, all these sorts of stuff. And I enjoyed that a lot. It's also a lot of work, of course. If you want to do that properly, it's a lot of work. And also finding a good German phrasing of all these papers is specifically also a lot of work in a way. It always feels a bit in very few characters indeed. Yeah, I mean, you can make these threads, so I just made threads.
Okay, yeah, of course, yeah.
And then actually most of the time I spent searching for figures that I would be able to use, because of course you can't use licensed figures from the actual papers. You can't just tweet them on Twitter. I don't think so. I think you need a license for it.
Though, right? Was it just people with their own papers? Or?
Yeah, I'm not 100% sure if that even is allowed. But I'm also not saying it's not allowed, but I didn't feel comfortable sharing anything that I don't have a license for. So actually I ended up just searching images that I would be able to use that the general audience would understand. That I could show my parents who do not have a neuroscience degree, and they would be able to understand. And I think that's important. In the end, this is where all this tax money is coming from. This is the society that we as a scientist also serve in a way. you overall, for a society to have basic research is great, and at least to some degree also luxurious, right? To be able to afford this kind of science. If you are a society that has a lot of problems, you end up cutting. Often these societies cut the science funding, sadly, because the money is just needed elsewhere, more immediately. Whereas basic science is often a long-term project that you need additional money for. And to explain why you need that and why it's important and why your research is important and why overall science should be funded and why science, also basic science, is critical. This depends on the acceptance and the motivation of society as a whole. Chipping in and funding all of this stuff. So I feel, this is important.
Yeah, I mean, to me, that's like, I mean, I think there's a few reasons why I want to do this podcast, but one of them is also that just, there's a lot of people who seem to be interested in these kind of topics and who, just have a completely different life and don't have the time to spend hours reading papers or whatever. And I think, most stuff I think can be communicated fairly easily if you try. At least I think, especially from psychology, because most of the stuff you study is to some extent about stuff people talk about anyway. I know, like, hexadirectional symmetry is a bit harder to explain using just words. But
the funny thing about that signal is if you see it, it's very clear, and if you see a grid cell firing rate map,
it's the most obvious.
You know what
thing.
it is. Yeah. It's very obvious, but putting it in words can be overly complicated. And also, I mean, you probably noticed in this podcast as well, is that I didn't find the best phrasing yet for grid cells. The most concise one. It's always like, oh, this is how it. But
I think, I don't know. Yeah, I've also wondered like how to. Yeah, I mean, maybe I think like one way might be to like link it to stuff that people know, right? Let's say like a honeycomb is a hexagonal pattern.
Mm-hmm.
I don't know how clearly people know that. It is a weird thing where I feel like if you just say like, just look at this photo, like then people will know. There must be like some good real life examples where you can just say like, it's like that.
Mm-hmm. There are, of course. Yeah. So maybe I can ask you a question. You already mentioned it a little bit, but so the motivation for you to establish this podcast. By the way, I find this amazing that you do that, because you mentioned before this recording, you also mentioned that this was a long-term dream or ambition of yours.
Yeah, I've been thinking about it for a while.
To start this,
So basically, I mean, I think I just always, I mean, one of the things is I just always like listening to podcasts. So I've been, I don't know, for 5 years or something. And I think that's at least when I became aware of podcasts. And I think before then, I listened to whatever, like long form interviews on TV or whatever, whenever they were available. And I don't know, I just always enjoyed listening to usually two people talk. I think in general, I'm not someone who's a huge fan of groups. I like a dialogue or like 2 or 3 people at most. And so I think I've just always enjoyed listening to people talk about something that they and I find interesting. And so, so I think, I don't know, I think in general, when I see someone do something that's cool, I just want to do it also, which is, I think, a very natural habit. I think I have it maybe more than others, and it leads to me starting like various different projects, which can be a bit overwhelming then sometimes. But so then I think for the last 4 or 5 years, I've been like vaguely thinking about doing something like this, but never really had the, I mean, one thing is, I think 4 or 5 years ago, I don't know how good the technology was to do this kind of stuff. I mean, you can use Skype and that kind of stuff, but I always felt like the audio quality there just wasn't that good. Maybe it was, I don't know, it seemed to me like that. And then, I mean, also I felt like I didn't quite have enough stability in sense of like knowing what I wanted to do. And like, like when you're doing your masters or whatever, and you move. I mean, I've been pretty much moving around like between cities almost every year for the last 10 years. No, well, not exactly. I had like 3 years of bachelor's, but apart from that, it's been like a new city every year. But I feel like now I have a bit more stability in the sense that, I know like I have like an income for a few years where I know like I can afford the software or something and the hosting. Yeah, I thought I'd just try it. I mean, another thing was also, as I said, one thing I think is, it's like science. I mean, I'm not like into science communication per se, but I do enjoy like popular science books about topics I know nothing about. But another thing is also just that, it happens to me so often, or to everyone probably, that who's a scientist, you read a paper, you think something's really cool, and you have a few questions about it. And then usually you just kind of have those questions and you try and answer them yourself, or you maybe ask someone who might know something about it. But I feel like in most cases, you don't just go up to the author and ask them. At least I don't, unless you happen to know them or something. So part of the hope is, part of the hope is that I can just talk to people whose stuff I find cool and like ask them about it, right? It's just, another thing is maybe, so we have these talks in Hamburg, and you know about it because you were one of the speakers. And then people, they usually come from somewhere else to Hamburg, then they stay like a day or two or something, I don't know. And then they give a talk at some point in the day, and then they usually have they'll send like an email around saying like, hey, this person's coming, does anyone want to talk to them? And and that's cool, and I've done that before, but it's always so limiting because you usually have like someone who's like spoken about the same things with like 4 people before you or something for the last few hours. They may be a bit tired or jet lagged or whatever, and then it's just not the most efficient way of doing that. So my hope is I'll just contact the people I want to talk to and then just learn something about their work and also how they approach stuff.
Yeah, it's amazing. Yeah, and if I understand you correctly, you also don't only invite scientists, right? It could be anybody.
Yeah,
that you think has something interesting to tell.
Yeah, I'm just interested in like loads of stuff, and so I read lots of books. I enjoy film, music, art, whatever, sports, and so there's just the hope is that I can also talk to those people about whatever they do. Right now, one reason why a lot of my guests will be neuroscientists and psychologists and whatever is because I know a few of them. For example, it was much easier to ask you because we've met before. Than to just like randomly mail someone who has no idea who I am and these kind of things. So I know a few people in science, and I think it's also easier for me to establish connections there because I can say like, hey, I read these 3 papers of yours, and I can really show like I know their stuff. So I think most guests probably will be from that, at least in the beginning. But yeah, the hope is, I have like a few guests already, or a few people who said like they were going to come on who are not scientists.
Maybe eventually you could invite somebody who is running a podcast and discuss basically setting up a podcast.
Yeah, right? I've already.
I think for people like yourself, people who are planning a podcast, that actually might be very interesting.
Yeah, it's. These meta things I've always found them slightly annoying.
Okay.
It could be fine, but you read a book about someone who's writing a book where the person in the book, it's like, okay, you're very clever, but but in this kind of non-fiction sense, I agree. And there is already one guy who, I'll not mention the name now, just because he might say no. And but there is one guy who I'm going to ask because he's doing a somewhat similar podcast. Yeah, because it's, just to see what his experience was like so far.
Yeah, it would be fun.
Yeah. It is a bit work putting up the podcast and that kind of stuff, but it's my hope at least is that it's not like a huge amount of work. We'll see. I mean, like right now I do read like, I read your Nature Neuroscience paper, your TICS paper for this. So that, that does require some effort.
Of course, yeah. And it makes sense to invite on people that you actually want to read the stuff.
Yeah, or you often I just have, right? Like in your case, I hadn't read the papers before, but I saw your talk. So, yeah. Do you want to start your own podcast?
No intentions here at this point. No,
Okay.
no time actually. Yeah.
Yeah, we'll see how the time thing works out. Yeah, I'm just starting it. We'll see. Yeah. I hope I don't like dig myself into a hole here. I'm not going to get out of. Okay, so I mean, you mentioned earlier is the plan then that you stay in Trondheim until the end of the year now, or maybe can you explain, like you wanted to go to the US, like you finished your PhD, you wanted to start a postdoc in the US in March or whenever it was. So when did you want to go there first?
Yeah, so in March was the initial plan. That was postponed to May. Then this was postponed to July, and now it was postponed to, I don't know, November. Who knows? Because of the COVID-19 pandemic, of course, it's really complicated making any plans at the moment. But yeah, so my initial plan was to start a postdoc at the NIH with Chris Baker, who's an expert on scene on high-level visual cortex as a whole. But I'm very interested in working with him for his work with on scene processing, because I think it really connects these low-level visual areas to these hippocampal cortices that I'm interested in. So this is the interface, if you want, between the two worlds. Also between my work with Andreas Bartels and my work with Christian Doeller, I feel like Chris Baker is right in the middle, bridging all of those levels in a way, bringing those together. And the plan was to start in March initially. Now it's almost August. So that's a bit frustrating, of course, but the plan still is to move there, and I'm really looking forward to that as well.
Yeah, I mean, it sounds like you're, in a sense, lucky that that Christian can support you for a bit longer, that kind of stuff, because I feel like there must be a lot of people who are in a similar situation to you right now and who aren't in that position. And then
absolutely, yeah. No, I'm also deeply grateful for that. Absolutely, yeah. And I mean, Christian and I, we also have quite a few projects that I would have worked on anyway, even after starting at the NIH. So now I just have some more time to actually get those further and maybe even finish some of those. And that's great in a way, right? So I feel like I'm making good use of that time now, but it was just not how I initially planned it. I mean, this entire world, entire 2020 for everybody, for all of us, was a mess and confusing, if you want. And it was also for me.
You're one of everyone.
Exactly. Yeah. I think the difference a little bit is that I still don't know when things are moving forward.
Who knows?
You mentioned that in Hamburg things become a bit more normal now, and also here in Trondheim things are back to normal. In fact, we already scanned here in Trondheim again.
Oh yeah, we've been scanning for
a
month or two or something. I can't remember. I mean, basically, I don't know exactly what the trajectory is, but I think for the last 2-3 weeks now, Hamburg has, on any given day, on a 7-week average, I think they had like 5, 6, 7 cases or something in Hamburg for the last 3 weeks. And Hamburg has 2 million people, so it's very limited right now in terms of the number of infections.
Mm-hmm.
I don't know, it always seems to be like a bit of an ebb and flow right now, where you have like individual outbreaks in Germany. But right now, I guess general, the northern part of Germany seems to be pretty, like Mecklenburg-Vorpommern didn't have a single case, I think, for like 2 weeks or something.
Mm-hmm.
But who knows how. I mean, I guess they're gradually opening stuff again, and then kind of see how that affects transmission rates. But yeah, so far I don't know whether everything. I think pretty much everything is open now in Hamburg.
Yeah, here in Trondheim too. I think things are almost back to normal. But then I always feel like I'm just waiting for something. I'm waiting to leave, and it's a bit taxing, I must say. And always this short-term postponing is taxing.
Like short-term, we're talking like 2 weeks before? It's like, nope, you're not coming, or is it sometimes been?
I still don't have the visa, and the embassy is still closed, and there's no information from the embassy when they will reopen, and they don't know. So I always end up waiting as long as I can before making any new decisions and new arrangements. But that also always means that really on short notice we just change the entire plan. And it's not a small plan, right? It's moving to the US. It's moving our entire lives to the US. And I've been planning this for so long now that it's about time to actually do it.
So like us means you and your wife, or?
Yeah, exactly.
So what is her situation? Like, does she, like work-wise or something, does she have, I don't know, does she have something there that she's going to do then there, or and it's the same thing as you, or how does that work?
Yeah, luckily she's working here at the hospital, and so in the medical genetics department as a tech, how you say, it's in Norwegian we call it bioengineer.
Okay, so, like an engineer, or
it's lab tech, if you want. Running all these analyses that can also be used for COVID-19 diagnosis. So she doesn't have a job right now in the US or yet, but the chances of her finding something are, I think, fairly high because of her background and the immediate application of basically her somewhere in the hospital there. And I will be working at the NIH.
Sounds like that.
Yes, so job. Exactly. It feels like we already know quite a few people who might be interested in recruiting her as well. Yeah.
And okay, so assuming you get there at some point, whatever that might be, do you already have like specific experiments you're gonna do, or is it still very much like, I don't know, you discuss a general direction with Chris Baker, and then you'll see, you'll figure out the details when you get there? Or...
No, I think the experiments are laid out, so I have plenty of ideas and discussed them with Chris also. Identified the ones that overlap with his interests most. And so these projects are, I think, some of the scripts are already done, even like stimuli and stuff.
Ready to go.
Also the lab, of course, already has a really a good foundation in scripts and a good collection of retinotopic mapping stimuli and analysis packages and so on. And also I myself have quite a few stimuli that I wrote and never used, and basically just adapted already quite a few scripts. And conceptually, these projects, I think, are laid out nicely already.
So it's kind of like
I'm just waiting to actually do it.
But you can't do them where you are now, or like, I don't know, like you have some sort of, I guess you say you're already starting other projects now with Christian. But let's say, I don't know, there's a second wave in the US in autumn, and then it's like you can't come in, or you don't want to go in, I don't know, for like another half a year or whatever. Is it like, could you just run the same stuff in Trondheim, and it's a collaboration with Christian Doeller's group then, or?
Yeah, so this is one of the plans for that project I mentioned that I'm starting with Christian. It's actually a project that we've been planning for quite a while, but then I thought I wouldn't have time before the US, before our move, to run it here in Trondheim. And now I might scan it at the NIH, and it will be a collaboration with Chris and Christian. And who knows, if, let's say, the US remains closed for the next year or so, then I might end up scanning it here, or maybe also in Leipzig. Christian is not flanked in Leipzig.